Okay, so I wanted to start by thanking you guys for organizing the symposium. It's really, really amazing. And I'm going to dive into a slightly different perspective of precision mapping and focus on task-based mapping of the functional networks in the brain. So I hope this presentation might give those of you guys some hope who are thinking like it's the only way forward, scanning people for 20 hours every day for a year to get something that's that allows us to really model the individual brain, idiosyncrasies. So I'm just going to quickly, it's actually, can I see the presenter view? Or not really? No, whatever, okay. So I'm going to start with some recapitulation of things that were already said. So, when you think about the ultimate goal of cognitive neuroscience is mapping the cognition, the cognitive functions to some neural implementation in the brain. And in this, we have to face a big challenge, which is related to the fact that brains are very different from one another. And this relates both to the structural differences between different brains. And this is a little bit less of a challenge nowadays because we have really good algorithms that allow us to like precisely register, to precisely map individual brains of like individual people, but when we do functional imaging, what we have to do is to stretch the signals that come from those very different brains of different sizes and different, very different brains in general, to something that's a common template. That's, as Tim already mentioned, that results in losing a lot of information that comes from individual differences between participants. But structural differences are not our biggest challenge in functional mapping of the brain. The biggest challenge is related to the fact that there are also huge functional differences in how our cortex is organized. So here I'm showing you an example. That's an example of the language system. And although you can see that different individual brains show more or less similar responses in terms of macroanatomical structures that are related to language processing, in individual subjects, there are some differences in how exactly within these macroanatomical structures the representation of language is organized. And we can think about these differences as fingerprints. So they have been shown to be very stable within participants between different tasks and across the lifespan. So what happens when we register that again, like to the common template, is we lose all of that information about individual differences. And these issues related to variability and function and structure of the brain are basically something that functional localization and functional precision mapping can deal with. So to just summarize the plan, I'm first going to go over what are the principles of functional localization using task-based MRI and how to implement them. Then we're going to talk a little bit about the advantages of functional localization, and in the end of my presentation, I'm going to show you an example of a study that we did to study the language system and the speech articulation system, and how can we use this precision functional localization to disentangle different functional systems in the brain. So let's start with how to do this. So the first thing we need to make the functional localization work is the functional localizer task. So this is a task that allows us to robustly localize a given process of interest in the brain. And in the case of language, what we do, we use a simple reading task, which contrasts reading sentences with reading non-words. And this simple contrast allows us to localize the brain regions that respond to retrieving meaning from words, like combining words into more complex representations and process the syntax of a given sentence. And most importantly, this is a very robust contrast that allows us to map the language system on individual brain, like in each individual brain of every individual participant. And crucially, the localizer task we use is more or less 10 minutes long. So you can do that if you're just interested in one process of interest, not mapping the general, all of networks, all of the different functional networks in the brain. What you need is to figure out a task that captures the process of interest, and then it doesn't have to be well, 20 sessions, a year of scanning or whatever. And what we do then with these brains, we treat these activations as a kind of masks, and then we can ask questions such as, how like the voxels in this mask respond to different type of processes. So for example, how do they respond to language itself, but also how do they respond to different types of mechanisms like domain general, visual, spatial memory task. And this way we can map, we can answer questions that are related to what is the role of the language system or like a different functional system of the brain, and precisely evaluate its response to different tasks while still keeping like the individual level functional, like individual level mapping in the brain. So, in a nutshell, the functional localization is a solution to the challenge of brain heterogeneity in a way that it allows us to model the individual variability at the individual subject level, but at the same time, it allows us to keep the analysis space constant between participants and between experiments, because it defines the analysis space functionally. So it kind of like flips the question. Instead of asking like, what does this brain region respond to in different people, we ask, what does the language system or the speech production system or the visual system responds to between different people? And this is also something that is ensured by using robust and similar localizer tasks between different experiments. So, now, does it mean that this is it? So not exactly, because there's a bunch of questions that are very difficult to answer if we only look at, well, the whole brain mask that we create based on a localizer task. So, for example, we can want to answer, like, ask questions like, are there any differences between left and right hemisphere processing in language? Or are there any differences between how frontal and temporal language regions respond to different tasks? Or we can also want to answer questions like, are there any differences between different kinds of tasks or cognitive or language-related tasks in a specific brain structure, like the left IFG or like some parts of temporal cortex? And to do that, we have to apply some kind of like group level constraints to our analysis. So one way of doing this is to use the anatomically defined constraints. So basically take an anatomical atlas and take a group level mask in the left IFG or the left SDS or like any other brain region that we're interested in. But there is also a way in which we can use the functional data from our task of interest, and based on that data, create a probabilistic map and parcellate that map into the regions of interest that basically represent not necessarily the functional parcellation that is at the same status as like an anatomical atlas or something, but they basically are very good in telling us there's a high probability that in a group of people you're looking at, this process is going to occur somewhere, more or less like this. And because this approach looks for the local maxima of activations, it allows us to parcellate the brain into different regions that fall into different parts of the brain and different hemispheres. Now, the question is, does it mean that we're leaving the individual subject localization at this stage? And the answer to the question is not, because the important part of this method is that within these group level constraints, no matter how we define them, we still define subject-specific regions of interest based on the functional localizer task. And we usually do that either by taking the voxels that respond a given, like above a given statistical threshold. So we take an anatomical mask in the left IFG, and we take all the voxels that respond like, I don't know, above a threshold of our choice. And this is the mask that we're going to use as the language mask in the left IFG. An alternative approach that our lab usually uses is to take some kind of like percentage of most responsive voxels. And the reason for that is because it provides us with masks that are like comparable in size between different participants. So that also is important if we analyze data from many different subjects. Okay, so moving on to the advantages of functional localization. I'm going to show you a couple of results from a study that we recently conducted on a large group of people in our lab. So unlike the studies that Laura was showing you, it's not a dense kind of like sampling of one participant. It's 772 people who all completed this 12-minute long language localizer task. And based on this sample, what we did is like we created this group level constraint, so like a probabilistic atlas, and then we parcellated that into different regions to then evaluate how do these regions on individual subject level, again, using functional localization, respond to different classes of tasks. So to look at their profiles, but also to what extent the language activations in individual people overlap with a couple of different standardly used parcellations of the brain, including the Harvard-Oxford Cortical Atlas, the DKT Atlas. The two of them are based on anatomical landmarks alone, and then the multimodal Glasser parcellation, which next to the anatomical constraints takes into account also information coming from the functional responses and functional connectivity. So I'm not going to talk about that second part a lot. The only thing I wanted to say that in a nutshell, we found that basically the anatomical boundaries in the brain are not exactly very good for mapping function, because basically functional responses in the brain and functional organization at the individual level does not always follow anatomical boundaries, and this is especially the case in the frontal cortex. I'm going to show you an example of the advantage of functional localization based on one of the anatomical parcels we used in the multimodal Glasser parcellation. And I'm going to show you how the profile of the responses to different tasks looks like if we take all voxels from that mask, which is something that people usually do when they do the ROI-based analysis. And how does it look like when we, within the same mask, define the most responsive voxels using the function localizer task. So the first example is going to be the left superior temporal sulcus, which is a brain region in this atlas that shows a very high overlap in all participants with language responses. So this is beyond doubt an area that is going to fall within the language system in most of our participants. So when we look at the responses of the entire ROI, we see that it robustly responds to language and shows very little, like actually no response to a non-linguistic control task that we had. However, when we look at the functionally defined ROI within that parcel, what we see is increase in statistical power. So this is a very important point for MRI studies, which oftentimes suffer from issues related to power. Using functional localization allows us to estimate our responses with much better precision. But it's not only the statistical power that's at stake. So if we look at the different ROI in the brain, which is the left inferior frontal gyrus, which is falls within the Broca's area. So this is a brain region that is usually associated with language, and it's used in many different studies as an anatomical mask to address different questions related to language and its relationship to different cognitive processes. So when we look at the response profile from the entire mask, what we see is that the left IFG shows a mixed profile. So it responds to language amodally, robustly in different tasks, but it also responds to a cognitive non-linguistic tasks. So the blue bars that you see here are the bars that correspond to a task where you basically have to memorize locations on the grid, so it has absolutely nothing to do with language. But if we define the analysis space based on our functional task, what we see is that the response to the non-linguistic task disappears completely. So basically, this example shows that functional localization provides a very powerful and very, easy to use in terms of how much time do you need to spend scanning, that allows us to disentangle between different functional systems lying side by side in the brain. So in brain areas like frontal cortex, which shows a very high variability in terms of functional and structural organization between participants, this is a very useful tool, because otherwise we're just risking conflating different signals and, falling into some false conclusions, let's say. Okay, so I'm not sure how much time do I have, because I don't see anything. Okay, yeah, so basically, we're not going to go into the speech example. Let me just quickly go, sorry. Happy to answer questions about this, by the way. Sorry. Okay. So the summary of what I wanted to tell you and convey today is that functional localization is, a relatively easy method to implement precision imaging on individual subject level, and it addresses the challenge of brain heterogeneity, but mapping the functional space of analysis if you're interested in one specific cognitive function. It allows us to increase the statistical power, and importantly, it allows us to precisely disentangle and characterize different functional systems in the brain. And here I would like to say thank you to everyone from EVLab at MIT who participated in data collection for this huge project that I was talking about, and also from other sites who were involved in the studies. Well, thank you.
We may have time for one very quick question. Here's a question from the chat. How well do these language localization findings generalize to natural language processing?
Natural language. Oh, okay, so basically, there is a very high correlation between the resting state networks in the brain, localized, and the task-based localization of language. So there are studies, for example, from Randy Buckner's and Rod Braga's group, where they show that if we use the very densely sampled functional resting state data, we retrieve a network that corresponds more or less to the frontotemporal language network. And at the individual participant level, we see that these networks are very highly correlated to the network that we retrieve with this 10-minute-long functional localizer task. So I would say that there is a pretty high, that it's a pretty precise method, even though it's a very simple task.
Thank you.
Okay.